For the next talk, we have Henry Chione from Waters, and he's going to talk about meeting the challenges of implementing accurate mass spec for biotherapeutic development in a regulated and non-regulated environment.
Good morning, everyone. Thank you for the introduction, and thank you for the invitation for us to talk in here. My name is Henry Chione. I'm a principal scientist from Waters Corporation, and this is my third time to give an oral presentation in the ASMS meeting, and I'm very excited, and I hope the third time is a charm. The title of my presentation is Meeting the Challenges of Implementing Accurate Mass Spec for Biotherapeutic Development in both regulated and non-regulated environment. Here's the outline of my presentation. I will start to describe some of the challenges, and then I will talk the BioAccord system, how it's designed and built to meet those challenges. I use the data to show the BioAccord workflow applications with basic performance data and case study data to show how it will help to address those challenges. And at the end, I will give a brief summary. This is a paper many people in the audience may know. It was published 2 years ago by Sarah and the team in FDA. And in the paper, they studied 80 BLAs from 2000 to 2015. And out of the 80, 79 of the filings used mass spec data. In addition, the number of attributes used by mass spec in the filing increasing, as shown on the right in here in the graph. In the paper, it stated that we expect that we will see additional MS methodology within the quality control and comparability sections. So there will be an evolution of LC-MS usage from non-regulated environment to highly regulated, high compliance environment in the industry, as we can see. From the user perspective, as we all know that there are LC-MS and LC-MS QC release assays is already in place, but very often it is not the first choice. The reason for that is the many challenges our users using. A few years back, when Waters started to think about to develop the next generation LC-MS systems, we did a survey with our user, and here are the top 5 requirements from our users: robustness, reproducibility, easy to learn and operate, small footprint, and informatics for compliance-ready and workflow-driven. With that, we went to work and we built the new system, the BioAccord system, which was launched in January WCPB this year. It's the first smart MS enabled by a former LC-MS solution. Now, it is not one component, it is a system. It consists of the LC I-class, Acuity I-class plus system, and the Acuity RDA detector. I'll talk a little more about that. And also, it's controlled by the compliance-ready unified informatics solution. The system is built and designed to optimize for robust performance, workflow-driven, easy of use and maintain, and the transformation engineering, that's what we call, really made the small footprint, as you see in the picture in here. It's next to the LC stack. It's very small, and it's compliance-ready to make sure the data integrity is taken care of. In Ben's presentation, he used the color display in here. The front panel of the BioAccord system, the RDA detector, also used the color display in here. The 3 colors will indicate the status of the system. Also, the system has a lot of self-diagnostic tools built into it. Therefore, the user in any level can be easily do the diagnostics and maximizing the uptime of the system and productivity. I'm going to show the schematics of the system, and just 2 points I want you to take from the slides. One is this is a this is a thermophile system. Although it does not have a quadrupole in the front, it does generate fragmentations that can be used for supporting OS investigation such that. In the development process for this system, we built into a program we call Beta Access. So we invite many of our users to come to the house, to our lab, and to run their own experiment with their own samples, their own workflow, to test the system. We want to make sure we'll make the fit-for-purpose system. Pfizer was one of the users collaborate with us with this effort. And this is the Pfizer's platform method workflow in supporting development and also manufacturing and QC. And I will show some of the samples from this collaboration as the case study sample. Before that, I'll show the basic performance of the system. Reproducibility is very important, as we see from requests from our users. This is the intra-system reproducibility experiment using the NISMAP intact standards as the sample. We're looking at the intact mass, the convoluted spectrum in here. We're monitoring the top 5 peaks. We run the experiment for 5.5 days with about 1,100 injections. As you can see, the relative abundance of those identified components automatically calculated within the unified system. And we have a very straight line in here showing, I hope you can see that the percentage RSD is very tight. We run the same experiment over a longer period of time. We run it in day 1, day 2, 1 week, 2 weeks, and 3 months. And for the same components, the relative abundance will still have very tight percentage RSD. How about amongst different systems? So we run the intra-system reproducibility as shown in here. We run the same experiment on 6 systems, displaying here 8 experiments. Again, we get very nice reproducibility showing percentage RSD is very tight. This experiment showing that system is reproducible inter and intra-systems and over a long period of time as well. This is one of the well-characterized sample from Pfizer, and we're looking at the trisulfide bond detection experiment. Trisulfide bond for the maps in here, excuse me, it happens between the light chain and heavy chain. And the goal is to detect the trisulfide bond in the intact level. As you can see from the spectrum display in here, we do see that from the BioAccord system with very good mass accuracy. Also, we calculate the trisulfide bond level, which is 29%, but the calculation is showing here, which is identical from the platform method. Another experiment we run is to detect trace level sequence variance in the subunit analysis. So this, again, very well characterized sample is ice digested and reduced, and we can see 3 fragments in here, and showing is the TUV chromatogram. On the top is the platform method, and the bottom is from the BioAccord system. Very comparable. The BioAccord system, we adjust the retention time to half of the run. What we're looking at here is really low level at 0.3% of the detection. Therefore, if you run LC-UV experiment, you won't be able to do the quantitation. So mass spec is required if we look at the 3 peaks in here and combine the spectrum, deconvolute spectrum, and from here, 1, 2, 3 comparison, you can see the detection of the 0.3% sequence variant for this experiment from the BioAccord system. ADC sample represents a more complicated molecule type. And for 16 conjugated ADCs, as we may know that we need to run native spec, mass spec, to do the intact dot calculation. On the left in here, I hope you can see that is the raw spectrum envelope for 3 conjugation level ADCs. And the middle is the zone in one stage, one single charge state of the spectrum, showing very nice distribution of glycosylation forms. Also, the conjugation level increases from the top to the bottom. And on the right is the deconvoluted spectrum with very nice pattern matching the raw spectrum. And on the right, here the number of automatic calculated numbers for different conjugation. And this, what's not showing in here is that the sample set we have run previously in multiple systems, and the LCMS data on the BioAccord is comparable to those experiment as well as to the Higgs separation experiment. So we'll move down to peptide mapping experiment. Reproducibility for peptide mapping, as previous speakers indicated, that is very important for MIM studies, for example. And we're looking at here is the reproducibility of the retention times. We run 18 injections of the NIST tryptic digest sample over about 24 hours. We overlaid the chromatogram in here, as you can see, they're really on top of each other. For peptide mapping experiment, we very often want to look at the modification of peptides. So here, we're going to pick a few glycosylation modified peptides, oxidation, terminal truncation, and deamidation. Look at those peptides, and then we'll plot the retention time across 3 different systems. So we run the same experiment on 3 systems, and you can see we get very straight line in here, very good retention time standard deviation on the right showing. Also, we can automatically calculate the relative abundance of the MS signal of those peptides and plot in the same way. And you can see we get very tight percentage RSD, indicating that the system is very good for peptide mapping and peptide monitoring kind of experiment. One of the test sample experiment we did when Pfizer came in was this degradation experiment comparison. So on the top is the control sample, on the bottom is a degraded sample. We're going to look at those 3 peptides. You can see the change with the star indicating in the chromatogram, mirror image in here. And if we look at one of them, and on the right, it shows the fragmentation spectrum that can be used to localize the modification site. And middle in here is extracted ion chromatogram, the peak area they used for the relative abundance calculation. We plot this and compare the experiment result with the LC-UV in-house release assay. As you can see that the BioAccord data compared very well with the release assay, and also it's comparable with the in-house MAM approach method using the Exactive platform. For the other 2 modified peptides, there are no in-house release assay, but the 2 systems, again, very comparable. Move to the release glycan workflow. For BioAccord system, we can run the experiment, automatically identify all the release glycans and quantify the release glycans. And the identification happened using the GU value from the chromatogram, as you're showing in here, and confirmed the identity by the mass spec data. After the run, you get very nice chromatogram of the fluorescence detector in here, and with all the label of the identified glycans. If we look at three levels of glycans in terms of relative abundance, showing in here, plot then our 20 different runs. And again, you can see that we get very nice percentage RSD showing the reproducibility of this workflow. In summary, the BioAccord system addresses many of the challenges of implementing accurate mass spec for biotherapeutic development with all the features listed in here. And case study experiment results are comparable to the in-house platform methods, and the BioAccord system is highly reproducible and for all the workflows, and we believe that it's ready for late stage and QC environment. I want to thank my coworkers in Waters. In the past 2 years with the project, we get a lot of help from folks from both the Milford, Massachusetts side and also from the Manchester side in UK, and a friend from Pfizer. And I'd like to recommend you, if you want to know more about the BioAccord system, we have 4 more posters presenting in this meeting, and MAM study for online IX, MS characterization of charge variance, and intact mass quantification study, and also release glycan of fusion protein and biosimilar study. With that, I want to thank you for your attention.
All right, we'll open it up for questions. I have a couple questions. So you showed the reproducibility for the intact measurement over 5 days. Was that the same sample you injected over that same 5 days? Or I'm asking because I'm curious about autosampler stability.
Yeah, it's a very good question. So what we did was we actually, we have made the NIST sample and as a standards. So we have different packages. And we use different packages for this run time, because you will not have enough to run for 5 days. So those are different samples.
Okay.
Hi, I have a quick question. It's my understanding that you're still using leucine and Keflin as your mass calibrant. And I was wondering, now that you're doing things that occur at much larger M over Z values, like native mass spectrometry, have you considered changing your calibration solution?
For this system, the answer is no. For this system, we run all kinds of experiments. As you can see up to the native mass spec, we don't see any issue for the mass range. That's first of all. Secondly, on the system level, as we're trying to make it a system, during the production process, we have a SIT process with system integrate test that will put all the LC component, mass spec component together and test before shipping to our customers. So for the mass accuracy is one of these areas we're looking at carefully. For all the SIC tests, we do not see any problem using the LuINC as the reference, as the calibration compounds in here. Yeah.
Thank you.
Yeah, you're welcome.
I have another question. When you were evaluating the glycopeptides, did you also look for in-source fragmentation induced by the system, or was that evaluated?
So as many of you know that when you run peptide mapping experiment, the glycopeptides is very fragile. Very often you see in-source fragmentation. And for the relative abundance of the glycopeptides, we did not consider the, Nini, you may correct me that the in-source fragmentation, we use the, mostly the charge state of the intact.
Yeah, okay.
But what I want to say is that, what I want to say is that the source is It's a QDA-like source, so the source is much softer than any other source we have, and we don't really see as extensive in-source fragmentation compared to other vendors. So.
Okay. I have one more question. So the peptide map sample that you injected over 24 hours, did you do any differential analysis to look for new peaks that showed up between the injections over that time?
For 24 hours? No, we didn't. Because for our approach at this stage is very that to evaluate the reproducibility, and we didn't, because it's a NIST map, so we know very well we didn't try to do that to see if we get some new peaks. However, it's something we can do because the samples, the data is still there, and we can go back to look at that. So, but that's not something we did at the time.
Okay. All right, if there are no more questions, let's thank Henry one more time.
Thank you.